Oh, go ahead.
Do you find the button? Okay. Okay. Michael sits alone at lunch. He has no friends at school. He is one out of every 68 children living with autism. Country music legend Glenn Campbell now performs with the use of cue cards. His Alzheimer's prevents him from knowing that his banjo player is his daughter. He is one of 44 million that is diagnosed with Alzheimer's. Janice is one of 51 million individuals living with schizophrenia, and her voices tell her that she is worthless. Cerebral organoids, recently discovered by researchers at the Institute of Molecular Biotechnology, will revolutionize our understanding of brain disorders and may provide the key to finding a successful cure for these disorders. We will first describe cerebral organoids. Next, we'll dive into how researchers are currently applying these, before lastly discussing some implications and limitations to what Oliver Bussell, a researcher at the University of Bonn, states are absolutely remarkable. Let's first discuss how cerebral organoids were discovered, and then cover their form and function. Cerebral organoids begin within a petri dish. In 2015, Dr. Madeline Lancaster, a researcher at the Institute of Molecular Biotechnology, unintentionally stumbled across these organoids. Researchers were growing neural stem cells in the lab, but something went awry. As Lancaster noted, rather than staying in two-dimensional form, these stem cells were developing into three-dimensional clumps, which were then sliding off the petri dish. But rather than ignoring them or throwing them away, Lancaster found them fascinating. She wanted to see what would happen if she allowed for them to continue growth. So, Lancaster began experimenting with them, manipulating these cells into new shapes on a flat plate. But there was a major challenge here. How was Lancaster going to keep the tissue at the center of the cell fed without the benefit of veins? According to Russ Triskelion in a February 2015 article entitled Brain Organoids, Lancaster decided to immerse each of these cells into a nutrient bath that is known to nurture cells. She would then periodically shake or spin these cells to keep them awash in cellular food. So what are cerebral organoids? According to Madeline Lancaster, they essentially resemble a brain in the first trimester stage. Cerebral organoids begin with a single skin cell. In March of 2015, Jürgen Noblick asserts that with the right biochemical prodding, which is the use of chemicals to encourage a type of growth, these skin cells will develop into an induced pluripotent cell, which is capable of taking on any form of cell. This manipulation creates clumps of tissue the size of small pearls, which are then immersed in a peach-colored liquid. The MIT Technology Review in 2015 reports that these cerebral organoids grow to roughly about 4 millimeters in diameter in approximately 2 months. So they are, essentially, little brains in a dish. We have just described the cerebral organoids. Next, let's look at some of the current applications. Cerebral organoids are helping to locate causes of brain disorders, as well as support new genetic and pharmaceutical treatments. These organoids are helping to reveal the causes of brain disorders. In 2015, researchers Rudolf Janisch at MIT and Gao Li Ming at Johns Hopkins used cerebral organoids to investigate the etiology of disorders such as autism, schizophrenia, and epilepsy. Since the growth of these organoids mirrors aspects of normal human brain development, if something were to go wrong, researchers could discover at what stage the error occurs, and then isolate potential causes and mechanisms of this disorder. One such study was done by Andrew Jackson at the University of Edinburgh in 2015. Taking skin cells from a patient with microcephaly, the team then created cerebral organoids that resemble the patient's brain. They then replaced a defective protein that is associated with the disorder, and created cerebral organoids that appeared partially cured. Organoids are also helping to focus on genetic treatments, work that was previously impossible. The MIT Technology Review in 2015 reports that companies are now seeking to collaborate with both Lancaster and Noblick on possible genetic treatments. Researchers can now see how networks of living human brain cells develop and function. This unprecedented level of transparency holds tremendous promise. For example, Researchers at Yale University in July of 2015 were able to fix neuronal imbalances in brain disorders by suppressing a single gene in individuals living with autism spectrum disorder. A February 2014 article entitled Advanced Drug Delivery View finds that these organoids will assist not only in drug application, but in drug discovery. The article also holds that these organoids will shorten the time between when compounds are being discovered and when compounds are being developed. The article also holds that since these organoids are biomimetic, meaning that they better simulate actual body functions, that it will increase the likelihood of the drug's effectiveness as well as they do in vivo, which is in a living organism, as well as they do in vitro, which is in a laboratory vessel. For example, in an October 2014 article entitled Drug Discovery Through Stem Cell-Based Organoid Models, it asserts that pharmaceuticals could now be applied to these organoids to quickly distinguish a drug's effectiveness against various kinds of cancer. The possibilities here are endless. We have just described the current applications of cerebral organoids. Next, let's look at some of the implications and limitations. Currently, cerebral organoids can only be utilized externally. But in the future, neurons may be able to be implanted internally and repair or restore damaged brains. We will be able to move beyond identifying causes of brain disorders and actually be able to fix brain disorders. For instance, those living with chemical imbalances. In July of 2015, Dr. Flora Vaccarino finds that it may be possible to treat those with chemical imbalances in living human brain cells via clinical intervention. Organoids are also helping to reduce the number of animal experimentations. In a 2010 The Baltimore Sun article, it reports that just one test involves up to 800 animals. In a 2013 The Scientist article, it reports that while we never may be able to truly replicate the beauty of animal genetics, we will be able to reduce the number of animals actually being tested on, especially those in toxicology and drug testing. The United States Department of Agriculture released a statistic that over 26 million animals are tested on a year just for toxicology testing. Reducing these numbers is a step in the right direction. As cerebral organoids have just been recently discovered, there are some limitations. In August of 2015, Dr. Zamil Cater reports that the proper organization between both the brain and the blood supply are not present in cerebral organoids, and this limits their size. In 2015, Dr. Manuel Casanova expressed reservation over the fact that researchers are now willing to model an entire human brain just off of cerebral organoids. The main concern here, though, is the fact that these organoids are created externally through biochemical prodding, not through the natural process of biology and development. Another large concern here, though, is the fact that cerebral organoids mimic a human brain, but one essential part of the brain is missing: the cerebellum, the part of the brain that handles motor skills and language. However, Lancaster reports that this isn't surprising, is the fact that the cerebellum develops after the first trimester stage. Until these organoids can be met, or until these setbacks can be met with time, funding, and breakthroughs, we will not see the true potential of cerebral organoids. Cerebral organoids hold the promise to revolutionize our understanding of brain disorders. We have first described cerebral organoids. Next, we dove into some current applications, before lastly discussing some implications and limitations to this truly remarkable discovery. Michael plays with his friends at lunch. Glenn Campbell hugs his daughter on stage. Janice knows she is worth something. Cerebral organoids, the discovery of today that can heal the minds of tomorrow.